Okay guys, so now we're ready to continue with our microscope lab. So we're gonna start right here on part 2 where it says eukaryotes, and we're gonna look at some animal cells today. So I'm gonna prepare a slide of a cheek cell, and yes, it's gonna be from my mouth, but you won't see me do this. So I'm gonna follow the instructions here. I'm gonna start with a small drop of water that I'm gonna place on my slide, and then after I place the water, I'm gonna perform part 2, which is I'm gonna use a toothpick and then scrape the side of my cheek on the inside and try to get a small amount of cheek cells off my cheek. And then I'm gonna transfer that to my slide. So I'm gonna go ahead and grab the water so you guys can see me do this part. I'll add the drop of water to the slide. And that's so when I transfer my cheek cells that I'm not just putting them on a dry slide. I want to try to get those into that drop of water. So now I have some cheek cells hopefully on my toothpick, and I'm going to kind of go back and forth in the water. See if I can transfer some of those cells. Now you don't see them because they're not stained yet. Okay, so I do that. I'm going to throw my toothpick in the trash when I'm done, but right now I'm just going to set it on the paper towel. Okay, if I go back to my directions, it wants me to add a small drop of methylene blue stain and then add a cover slip. So methylene blue is really dark. That's why we want to add a small drop because sometimes if it's too dark, you won't be able to see the cheek cells. So I'm going to add a really tiny drop to that. There we go. And I want to now add my cover slip that I have ready right here. I'll go at a 90-degree angle so it will drop nicely. There it is. Look how beautiful that is. Now there's some excess coming out around the cover slip, so I'm probably going to dab that with a paper towel. So now my slide is prepared. This is my wet mount slide, and I'm ready to use my microscope. Just like the last time, I'm going to start off on low power. That's 10x. Make sure that's clicked in. I want to use my coarse adjustment knob. Make sure my stage is all the way down, and I'm going to use the stage clips to kind of make sure that my slide is held in place. There we go. And I'm going to go back like I showed you on the last one, use my mechanical stage knobs and make sure that this is centered above the field of view so that I get the best view ever. And I'm ready now to focus. I'm gonna use the coarse adjustment knob and bring it up until I get something in my view. So I have the cheek cells focused on low power on 10x. I'm gonna go ahead and let you guys see what it looks like through the eyepiece, but you do not wanna make your drawings on 10x. We wanna draw them on 40 or 400, but I wanted you guys to be able to see on 10x, if I could focus this, hang on, just give me a sec. This is very hard to do. Okay, this is through the microscope at 10x, but all you see are a bunch of little blue circles everywhere. You don't see it really clearly, and so I'm gonna put this on 40x or 400 times to give you guys a better perspective. So hang on, I'll do that. Okay, so I have it focused on 40 or 400 times. I have a pretty good specimen here at the end of my pointer, if I could focus this for you guys. Right at the end of the black line, push pause so you can see that. There you go. That is one cheek cell right there. And if you look closely, that dark dot that you see in the center, that's the nucleus. Tell me that's not cool. Look at that. That is one of Miss Lynam's cheek cells, and that dark dot in the very center is the nucleus. That's so cool. You'll notice they're not perfectly round. Everybody thinks animal cells are perfectly round, but they're not. So that's what you guys want to draw, is that one cheek cell. You want to draw that in your next field of view on your answer sheet. Okay, for our next specimen, we're ready to move on now. We're going to go to part 2, eukaryotes, and observe what we see in pond water. So I've already added a small little drop of pond water to my microscope slide. I've cleaned off the methylene blue from the cheek cells, and I'm ready to put a cover slip on this and then see if we can find anything under the microscope from the pond water. So I'm going to follow the directions, carefully add my cover slip at a 45-degree angle so that I can, not kill anything that's on there in case I have some amoeba or paramecium. And then I'm going to observe, look at step 4 right here, observe for different types of organisms, and then draw what I see on my answer document of anything that I might see. So I'm going to see if I can find some stuff for y'all. Okay, so for the pond water, I would like you guys to use this amoeba picture. So this is what you're gonna draw for the pond water. I did see a lot of things under there, but it's kind of hard for me to identify for you. So go ahead and use, this is a typical picture of an amoeba. You can see the nucleus, that darker kind of pinkish color in the middle. So that's gonna be your pond water specimen. So for the last specimen we're going to look at, it's actually going to be a comparison. This is part 3. It's a comparison of bacteria, the prokaryotes, to all the other eukaryotic specimens that we've observed. So this is going to be really cool because bacteria are tiny. Even the largest bacteria is hard to see on the microscope. So I'm going to try to do a good job of focusing this for you guys. So read the paragraph here. Make sure you look over that. And then you're not going to be able to see anything inside the bacteria. You're just going to be able to see the shape of the typical bacteria. So I've got a prepared slide right here. If I hold it over paper, you can see that purplish area. That's where the bacteria are on the slide. So I'm going to take this and focus it on the microscope, just like I showed you guys, starting off on low power with my stage all the way down. I'm going to secure my slide in place like we do all of them. And then use my mechanical stage knobs to make sure that this is centered. I want to see if I can get the purple area over the light. There we go. And now I'm ready to focus on my 10x. Okay, so I have it focused on 10x or 100 times the original size. So if I can focus in here, give me a second. I'm going to show you. Look at all these bacteria. Hang on. This is a lot harder than you think. Okay, there we go. Check that out. That is like millions of bacteria. You don't want to make your drawing from this view. This is way too many. I'm going to see if I can focus it on 40 to get 400 times the size. So give me a second and I will do that. I'm going to go ahead and click it in and then use the fine adjustment knob to see if I can find it a little bit better. Okay, so this is bacteria on 400 times or 40x. Look at this. Ooh, that's pretty good right there. Hang on. You're gonna probably need to push pause as soon as I get it. There you go. Now, hang on, I'm gonna tell you what to do. Let me try to get it for a little bit longer. There you go. Sorry if it's making you dizzy. There. Okay, what you're gonna wanna do for this one, obviously push pause so that you can see an area of this without me. I am very shaky right now. But do you see how the whole entire field of view was filled up? You just need to choose like a small area and make it a little bit bigger. The problem is it's hard to see the individual cells. So what I'm actually gonna do for y'all, I am going to go get some oil because this special lens right here that I use for my AP Bio kids, this special lens is actually a 100, which means I can make this 1,000 times the original size. So I'm gonna go get some oil so I can show it to you guys and maybe you can make your drawing and see some cells a little bit bigger. So hang on, let me go look. Okay, so you guys get a little bonus edition. See, it pays to be virtual, right? I'm going to use, I found the oil. This is Immersion Oil Immersion. And it's used for bacteria in AP Bio. So see how I'm gonna move that halfway? I'm gonna add a drop of oil. And what this oil does is it cushions the lens so it glides through the oil. So you can see I have the oil. Watch what happens when I take this lens. It's gonna glide right through the oil. Okay, let me see if I can get it right here. See that? Isn't that cool? So I just, I drug the lens through the oil. So now if I look at it on 1,000, I should get a better view. Wait until you see this. You're not gonna believe it. Go ahead and make your drawings on this if you want to. This is so much better. I cannot wait to show you guys. Okay, now I'm very shaky. Hang on, I'm gonna get this better. I'm gonna hold the microscope. There we go. Look at that. That is so much better. You're like, really, Miss Lanham? I can't even see it. Hang on, let me see if I can. There you go. That's pretty cool. It doesn't come out nearly as well on the phone as it is in real life. Okay, there it is a little bit better. So you can just draw a little area of that for your lab. Okay? So now you guys can go ahead and finish your lab. Make sure you have all of your drawings on the front side and on the back. Go ahead and fill in all the vocabulary and make sure you label the picture of the bacteria at the bottom. Okay, my microscope is put up. You can see it's right there. It's back on low power, stage is all the way down, cord is wrapped around the base. And that concludes our microscopic view of prokaryotic and eukaryotic cells. Hope you enjoyed it.